Hey guys, it's Medicosis Perfectionalis, where medicine makes perfect sense. Let's continue my endocrinology playlist. It's time to talk about hormones biochemistry. Simple concepts with huge implications. With that said, now let's get started. Please watch these videos in order, and this is video number 6. The facts explained in this video can be understood even by a freaking child. However, they have huge clinical significance. I will try a new numerical system. Okay, so we will do this. We'll start by a Roman numeral denoting the playlist. So endocrinology will have number 1. This is video number 6, so we'll use number 6. And then the idea or the fact will take number 1, 2, 3, 4, etc. And then you have A and B. A is for the analogy, the story, for the young kids. And B is the basic facts if you want to understand medicine. If you understand the analogy, you will get the basic fact. And then at the end of each video, try to think how many facts did we include and try to recall because it trains your memory. Analogy number 1. Any company has what Jordan Peterson might say a dominance hierarchy, starting by the chief executive officer, then you have general managers, and then you have two groups of people. The employees, they are hired by the company, they are part of the staff, they have to obey every commandment, and the independent contractors. Your endocrine system is the same. The CEO is the hypothalamus, the pituitary is your general manager. And then you have the employees, thyroid, adrenal cortex, and gonads. These are the people who obey the pituitary, especially the anterior pituitary. And then we have 3 people who could not care less about the pituitary. And these are the parathyroid, adrenal medulla, and pancreas. Analogy number 2. Okay, which one of these 2 activities is most important or most urgent for survival of the individual organism? You running from a tiger, or you participating in coitus? I gotta keep it G-rated for the algorithm. Of course, running from a tiger, of course, this is more important. This is more urgent. When you're running from a tiger, your adrenal medulla secretes epinephrine and nor-epinephrine. And that's why your adrenal medulla has to be an independent contractor, because it's important for my survival. But imagine that the adrenal medulla had to wait for the hypothalamus to secrete a hormone into the bloodstream, into the hypothalamo-hypophyseal portal circulation, and then goes to the pituitary to influence the pituitary to secrete a second hormone, and then the second hormone will be secreted into the bloodstream. It will flow in the bloodstream, and then leave its plasma protein, and then jump on the receptor, and after this I will act. The tiger would have eliminated your butt. But for intercourse, you can wait. Analogy number 3, the classic school experiment. Water and oil. Of course, water and oil do not mix, as a famous Egyptian president once said. I also reminisce about the story that they used to tell you in school. Remember, you get 1 stick. Okay, try to break it. Oh, easy, I can break it. But then, if we make a bundle of sticks, you will not be able to break it. Until your teacher gives this bundle of sticks to a very muscular student, the muscular student will break all of them at once, and then the teacher will say, the moral of the story is, as long as this doofus is with you, you'll be okay. Hormones are the same thing. You have lipid-soluble and water-soluble. Lipid-soluble or fat-soluble, of course, are water-insoluble. No kidding. Water-soluble are lipid-insoluble. We call those non-polar and those polar. Remember hydrochloric acid, for example. Once you put it in water, it's going to give you hydrogen, positive, and chloride, negative. This is polarity. You have a positive pole and a negative pole. Put some oil in water, it does not dissociate into positives or negatives. Why do I care? Let me finish. Your blood is watery, but your cell membrane is fatty. Oh, yeah, really. Your blood, is made of plasma. Yeah, and the plasma is basically water. Water is 2/3 of your human body. The cell membrane, however, is fatty. It was made of phospholipid. It was a lipid bilayer. Let me remind you, your blood had plasma and cells. The plasma had water and protein, such as albumin and globulin. Your blood is watery. However, your cell membrane is oily. It's as oily as the index finger of a junkyard mechanic. It has tons of lipids, phospholipid, cholesterol, and other lipids such as sphingolipids. Analogy number 4 comes from A Tale of Two Cities by Charles Dickens. It was the best of times, it was the worst of times. It was the age of wisdom, it was the age of foolishness. It was the epoch of belief, it was the epoch of incredulity. It was the season of light, it was the season of darkness. Oh, I wish I was born in Boston. It was the spring of hope, it was the winter of despair. In endocrinology, you have a tale of two hormones, the lipid-soluble and the water. They are different. Let me explain. Lipid-soluble, okay? I'm lipid, I'm fatty. Your blood is made of water. Therefore, I cannot float in the blood because this is lipid and the blood is water. They do not mix. However, you can carry me on a plasma protein while I'm in the blood. But once I leave the blood and go into the cell membrane is fatty. It's oily as me. Therefore, I can just squeeze through it, simple diffusion. On the other hand, water-soluble hormones, they are watery, and your blood is watery, therefore there is no need for plasma protein. They are freely floating in the bloodstream. But once they reach the cell, they will not be able to diffuse. Oh, because the cell membrane is fatty, but this is water-soluble. Therefore, you have to put the receptor on the outside surface, not on the inside, because it will not be able to enter. The receptor has to be waiting on the outside. So let's compare between lipid-soluble hormones and water-soluble hormones. Everything was lipid-soluble, everything was water-soluble. It was bound to plasma proteins. It was freely floating in the serum. Its receptor was internal. Its receptor was external. It was a genomic action, meaning since I was able to enter into the cell, I will influence DNA, RNA, protein synthesis, etc. But this one, the receptor is on the outside. This is a non-genomic action, has nothing to do with genes. It was slowly acting, yeah, because I have to be carried on a plasma protein. And then once I reach the cell, I have to leave the plasma protein. And then I have to diffuse through the membrane, which takes a lot of time. I'm slowly acting, but water-soluble is rapidly acting. So which way of learning is better? This way or your woke professor reading a PowerPoint? If you remember my biochemistry video on vitamin D, I've told you about the zinc finger. Vitamin D is a fat-soluble vitamin. Therefore, it will be able to pass and diffuse through the cell membrane via simple diffusion. And therefore, the receptor is not on the outside, it's on the inside. Could be on the nucleus or in the cytoplasm. Alright, and then once it binds to the receptor, you have the genomic action. You have some genes, and we have VDR, vitamin D receptor, enhancer elements, and this modulates gene expression. You have transcription and translation. Transcription is to make RNA. Translation is to make proteins. Analogy number 5. Imagine that you are an engineer working for a utility company, and you decided to turn the electricity off in this house, like they did in Texas. Terrible joke, I'm sorry. Which method is faster? For the engineer to just flip a switch, or for the engineer to leave his office at the utility company and then drives his car on the highway and then goes to your house, knocks on your door, tips his hat, kisses your puppy and goes to town, climbs over the ladder that you got from Home Depot using your credit card because you could not afford it, you cheap bastard, and then unscrew the light bulb? Which one is faster? Of course, flipping a switch is faster. Childish as this might seem, it's very important for endocrinology because water-soluble, they just flip a switch once they bind to the receptor that's on the outside surface. They just flip a switch. It is a rapidly acting hormone. But a lipid-soluble hormone has to wait until it unloads from the plasma protein, and then it goes to the cell membrane, touches the membrane, and then diffuses through the membrane, which takes time. This is fat inside of fat. And then diffuse to the cytoplasm, and then try to find the receptor. The receptor could be in the cytoplasm or could be on the nucleus. Of course, this is slow acting. And that's why when you're running from a tiger, running for your life, your adrenal medulla had to not obey the pituitary. It had to secrete a water-soluble hormone known as epinephrine or nor-epinephrine so that we can just flip a switch. But if you want to participate in coitus, take your time, take it slow. #serendipity. And that's why we say that lipid-soluble hormones have a genomic action because they influence gene, because the receptor is on the nucleus or in the cytoplasm. We will influence and stimulate gene expression, and then DNA transcription, and then into RNA, and then translation or protein synthesis from the RNA. However, water-soluble, they just flip a switch. It's a non-genomic action. They do not synthesize new proteins. They just release previously synthesized proteins. That's why they are rapidly acting. So where would you put the receptor? Would you put it on the cell surface? Would you put it in the cytoplasm or on the nucleus? Well, it depends. If you're water-soluble, you will not be able to enter. Therefore, it has to be on the outside. If you are lipid-soluble, however, welcome. Come on in. I put it in the cytoplasm or the nucleus. Here is another story. The story of the key and the lock. The truck and the dock. The ligand and its receptor. A specific ligand has a specific receptor. The ligand on its own is inactive. The receptor by itself is also inactive. Only the ligand-receptor complex is active. Only when the key goes deep inside the lock, only then is it active. Only when the truck hits the dock can you start shipping. So the ligand-receptor complex is active. Active to do what? To open a channel and hire Piers Morgan, activate an enzyme, or increase intracellular calcium concentration. Okay, this is normal. What is pathology? Well, pathology is a disease such as diabetes insipidus, diabetes mellitus. And what's the problem here? Well, the problem could be here, and we call this type 1 diabetes mellitus or central diabetes insipidus, or the pathology could be here in the receptor, and we call this type 2 diabetes or nephrogenic diabetes insipidus. Pathology can hit your ligand or it can hit your receptor. Either way, you will be inactive. And this is the definition of disease. There is no more ease. So in medicine, whenever you hear about type 1 or type 2, usually here is what we mean. Type 1, the problem is in the hormone. Type 2, the pathology is in the receptor. Example, diabetes. Type 1, no insulin. The hormone is gone. How about type 2? Well, there is some insulin, but the receptor is not listening. The problem is in the receptor called resistant or insensitivity. Diabetes insipidus. We have type 1 or central, no ADH. How about type 2? Well, there is ADH, it's just the kidney's receptor is not responding. How about rickets? Type 1, there is no enzyme, there is no 1-alpha hydroxylase. Type 2, receptor resistant. And then we have a similar situation with platelet problem. Decreased number, decreased function. Hemophilia A, decreased number of the factor, decreased function of the factor. TTP, decreased number, decreased function. How about leukotrienes? How do you treat asthma? You treat asthma by decreasing the number of leukotrienes or decreasing the function. You can either inhibit the secretion of leukotrienes or inhibit the receptors of leukotrienes. The first is called xylutin, the second medication is called the montelukast. And we have talked about this before in my pulmonology playlist. Implications. Alright, remember the CEO and the general manager, and we have 3 employees that listen to the pituitary and the hypothalamus. Okay. These hormones that listen, of course, are slower. Yeah, because we have to wait. And since they are slower, they are fat-soluble. Yes, because lipid-soluble hormones are slow. Lipid-soluble hormones have to wait until they unload from the plasma protein, until they diffuse through the cell membrane. But water-soluble do not. Fat-soluble hormones are carried on plasma protein. Why? Because they are fat-soluble and your blood is made of water. You cannot float in the blood if you're made of fat. I have to carry you on a plasma protein. Which type? Could be albumin, could be globulin. And that's why we talk about thyroid-binding globulin in the lab, cortisol-binding globulin, estrogen-binding globulin, or testosterone-binding globulin, or you can call them collectively sex-hormone-binding globulin. If you understood this lecture so far, this is going to be a piece of cake. The reason why you don't understand medicine is that your foundation is shaky. Conversely, have you ever heard of a parathyroid-binding globulin, epinephrine-binding globulin, or insulin? They do not exist. Why not? Because they are water-soluble. They are faster. These are the independent contractors. And here is a beautiful table that can help you cruise through endocrinology like a sharp knife in warm butter. Lipid-soluble versus water-soluble hormones. Lipid-soluble, okay, what are they? They are subordinate to the anti-pituitary. They listen and obey. But polar pituitary, who cares? Lipid-soluble, can you give me an example of glands that secrete these lipid-soluble hormones? Sure. Thyroid, suprarenal cortex, and 6 glands or gonads. Okay, what do they secrete? Thyroid gland will secrete the thyroid hormone, or what I call the stove of the body, because everything will be on fire. It increases your metabolism, it increases your heart rate, it increases your gastric motility, bowel motility, everything. What does the suprarenal cortex secrete? The corticosteroids, especially cortisol. How about the sex glands or the gonads? They secrete sex hormones so that you can participate in coitus. These are good times right there. On the other hand, the glands that secrete polar hormones include parathyroid gland, adrenal medulla, not cortex, medulla, and the pancreas. What does the parathyroid gland secrete? Parathyroid hormone. How about adrenal medulla, epinephrine, nor-epinephrine, etc. Pancreas, insulin, glucagon, and somatostatin. Next, what's the nature of these lipid-soluble hormones? Well, they are lipid-soluble. They are steroids and thyroxine. These are collectively lumped together. How about the polar hormones? These are proteins, polypeptides, peptides, and catecholamines. We lump them all together. Okay, solubility. I am lipid-soluble, of course. How about these ones? They are water-soluble. Therefore, I cannot float in the serum because the serum is watery and I am oily. You have to carry me on plasma protein, but now I'm polar. I can float freely in the blood. You can say P for passive. Since you are lipid soluble, where is your receptor? Well, I will enter into the cell by diffusing through the cell membrane. Therefore, you can put my receptor in the cytoplasm or the nucleus. Okay, how about you, polar? You have to put it on the outside because I cannot enter because I am water soluble. Action. Alright, I'm lipid soluble. I'm carried on plasma protein. I will enter the cell by passing through the cell membrane via simple diffusion, and I will synthesize new proteins by expression, transcription, translation. This is a slow process. Conversely, water-soluble hormones, the receptor has to be on the outer surface of the cell membrane. Okay, there is no synthesis needed of new protein. You just release previously made proteins, and this is a rapidly acting hormone. Let's look at it in another way. Lipid-soluble hormones are steroids and thyroxine. Okay, they are corticosteroids and sex hormones. All right, where did thyroxine come from? It came from an amino acid called tyrosine. Okay, thank you. How about polar hormones? You have proteins and peptides and catecholamines. Awesome. They come from the pituitary, so most of the hormones that come from the pituitary are water-soluble. How about the pancreas? Water-soluble. Parathyroid, water-soluble. Where did the catecholamine come from? Oh, the catecholamines came from the same amino acid tyrosine. So wait a second. One amino acid can give rise to thyroxine, which is with this group, and catecholamines with the water-soluble group? Absolutely, and this is important. These are called tyrosine derivatives. Story number 6. Are you bound or are you free? Which one is more active? The free. Lipid-soluble. I cannot float freely in the plasma because the plasma is watery and I am oily. You have to bound me to plasma protein. Since I am bound, I'm inactive. Therefore, I cannot readily or directly bind to receptors because I have to unload first. I have to unlock from the plasma protein and then I can go to my receptors. So therefore, I'm slowly acting. Since I'm bound to plasma proteins, this will make me big. Oh yeah, we are big. We are huge. We are not filtered by the glomerulus and therefore we are not lost in the urine most of the time. Polar hormones, on the other hand, freely floating in the serum. We are active. We are rapidly acting. We are small in size and we could be lost in the pee. Okay, Medicosis, let me ask about cortisol. Alright, sure. In Cushing disease, I can measure cortisol in the urine. And you said that all of the cortisol is bound to the... No, I did not say all of the cortisol. Most of the cortisol is bound to the plasma protein and will not be filtered. About 10% or less is freely floating in the blood. However, in Cushing disease, this 10% is greater than normal. It is greater in absolute amount and in relative amount. So, you will eventually see lots of cortisol in the urine. Let's talk about lipid-soluble. Okay, about 10% freely floating, and then the majority are on the plasma protein. But once I reach the membrane, I will unload and then diffuse. This is simple diffusion. I go inside, my receptor is in the cytoplasm or on the nucleus. Ligand-receptor complex or hormone receptor complex, now I'm active. I will stimulate hormone response elements or HREs, which will stimulate DNA, you have gene transcription, translation, all of this beautiful stuff, to synthesize new proteins from scratch. How about the water-soluble? I will not be able to pass through the membrane by simple diffusion. It's not going to happen. You have to put the receptor on the outside. Okay, and this will be the story of the G protein. G protein is the switch, if you remember the engineer. G protein connects the receptor on the outside with the enzymes on the inside. And this will be the story of the next video. We'll talk about all of the first messengers and second messengers. It's going to be epic. If you like this video, you will adore the mother of all courses, my antibiotics course. It has 40 videos and questions and cases and notes. You can download it today at medicosisperfectionalis.com. You download it once and keep it for you forever. Also, you can devour my new acid-base imbalance course. Talk about normal kidney physiology, diuretics pharmacology, and all of the clinical medicines and the anion gap and the osmolar gap and the serum anion gap, urine anion gap, base deficit, base excess, all of this crazy stuff, and even the Henderson-Hasselbalch equation. And on my website, you can get a 30% discount. Just use discount code SAVE30, available for a limited number of students only. Thank you for watching. Please subscribe, hit the bell, and click on the join button. You can support me here or here. Go to my website, download my premium courses. Thank you for watching. As always, be safe, stay happy, study hard. This is Medicosis Perfectionalis, where medicine makes perfect sense.